Hello, my name is Matt Austin. I'm an MD PhD student in Dr. Amelia Linneman's lab at the Indiana University School of Medicine. And I'd like to start by thanking Sugar Science for the opportunity to present our research. The title of my presentation for today is Dysfunctional Beta Cell Autophagy Induces Beta Cell Stress and Enhances Islet Immunogenicity. The primary focus of the Linneman lab is understanding how cellular stress response pathways contribute to maintaining beta cell homeostasis and survival, and inversely understanding how maladaptive stress responses contribute to beta cell loss and diabetes development. It has been shown that beta cell endoplasmic reticulum stress precedes hyperglycemia and type 1 diabetes development in the non-mouse model. Furthermore, there is observed downregulation of proteins and pathways involved in mitigating beta cell stress prior to diabetes development. This was observed by the downregulation of these pathways detected by mass spectrometry and longitudinal samples and isolated islets from non-mice prior to diabetes development. ER stress has also been shown to enhance production of neoantigens, including hybrid insulin peptides. These are just a few examples of accumulating evidence that suggests beta cell stress pathways play an important role in activating and or exacerbating the immune response in disease development. This has led to increased interest in how these stress pathways affect the beta cell immune cell interface. Our lab was the first to show the cellular stress response pathways of autophagy is impaired in beta cells in the development of diabetes. In these fluorescence images, LC3 is used as a marker for autophagosomes, shown in green, and LAMP1 is used as a marker for lysosomes, shown in red, as shown in the diagram on the right. Co-localization of these signals indicates fusion of lysosome and autophagosome for degradation of autophagy cargo. Thus, decreased co-localization corresponds with impairment of these latter stages of autophagy. So we come back to these images of islets from control autoantibody positive and individuals with type 1 diabetes on the left. When quantified, there was significant reduction in LC3 LAMP1 co-localization in the control compared to islets from people with type 1 diabetes. There also appeared to be an intermediary reduction in the autoantibody positive individuals. Additionally, we evaluated for the presence of telolysosomes and beta cells from donor pancreatic tissue sections. On the right are representative electron micrograph images. Telolysosomes are lysosomes that contain indigestible material such as highly oxidized and or cross-linked proteins. Oxidative stress and decreased lysosome enzyme activity can lead to accumulation of telolysosomes. We found that beta cells in the autoantibody positive group had an increased number of telolysosomes per cell, which could be indicative of impairments in autophagic degradation. Overall, our data show that the autophagy pathway is impaired in the setting of type 1 diabetes. Importantly, this pathway is impaired prior to the clinical onset of type 1, implicating this pathway in disease pathogenesis. Here's a brief background and overview of the autophagy pathway. Autophagy is a cellular recycling system that is stimulated under conditions of stress. Autophagy stimulus causes nucleation of a lipid phagophore, which binds cellular proteins and organelles, including damaged structures. It matures into an autophagosome, which then fuses with a lysosome to degrade its cargo and recycle the components. In addition to our data that I referenced, there are some other connections between defective autophagy and type 1 diabetes, and I'll highlight a few of interest. First, several gene variants associated with a heightened risk for developing diabetes are involved in autophagy, including cathepsin H, which is a lysosome cysteine proteinase involved in protein degradation, and C-type lectin domain containing 16A, involved in a specialized form of autophagy that is necessary for mitochondrial quality control. In addition, HLA class 1 islet hyperexpression is a well-characterized hallmark of type 1 diabetes, and defective autophagy has been shown to increase HLA class 1 expression in various cell types. Another connection to type 1 diabetes and autophagy is that autophagy is impaired by enteroviral infection in pancreatic islet cells, which is one of the hypothesized triggering events in type 1 diabetes development. As shown in panel C, treatment with low glucose stimulates autophagy compared to control, as shown by LC3 signal overlap with lysos tracker signal, as would be expected in the low glucose treatment, whereas E16 infected human islet cells show decreased overlap, indicative of impaired autophagy. Lastly, I would like to highlight the connection between hybrid insulin peptides and the autophagy pathway. Hybrid insulin peptides are an insulin peptide fragment linked with a fragment of another protein co-secreted with insulin, such as chromogranin A. These hybrid peptides are neoantigens that are recognized by autoreactive T cells in type 1 diabetes. Importantly, evidence suggests that the autophagy pathways are an important source of HIP formation. Taken together, these various pieces of evidence lead us to hypothesize that dysfunctional beta cell autophagy contributes to autoimmune targeting of beta cells by altering protein degradation and antigen production, leading to a change in beta cell immune cell dialogue. To better understand the role of autophagy in beta cell health and survival and whether defects in autophagy render islets more immunogenic, we're using a beta cell specific autophagy related gene 7 or ATG7 knockout model, which I will refer to as ATG7 delta beta cell. ATG7 is an essential autophagy enzyme, and thus the beta cells in these animals are rendered severely autophagy deficient. The diagram on the right shows that ATG7 acts in LC3 activation and lipid complex assembly and phagophore maturation in the autophagy pathway. We first wanted to characterize the phenotype of this model by tracking the mice blood glucose levels. And what we found, looking here at the Kaplan-Meier diabetes-free curve, was that all ATG7 delta beta cell mice developed diabetes at a sex-dependent rate, with males developing it at a median age of 11 weeks and females developing it at a median age of 15 weeks. After finding that these mice developed diabetes, we wanted to better understand the mechanisms by which the ATG7 delta beta cell knockout led to diabetes development. So we isolated islets several weeks prior to diabetes development, and bulk mRNA sequencing and proteomics was performed. The top upregulated pathways at the transcript level are shown here on the left. Some pathways of interest that I will highlight is an upregulation in the inflammatory response and immune system process. Upregulation of these pathways suggested potential enhanced immune cell recruitment to ATG7 delta beta cell islets. The upregulated transcripts associated with these pathways are displayed in the heat map on the right. We also observed upregulation of transcript involved in tumor necrosis factor and IL-6 production, cytokines that have been linked to early events in type 1 diabetes development, including immune cell recruitment and activation. Now looking at the pathways that were downregulated at the transcript level, we saw several pathways associated with cholesterol and lipid biosynthesis and metabolism. Evidence from the literature suggests ER stress negatively regulates cholesterol export and metabolism, which could explain the downregulation of transcript involved in these pathways. There is also downregulation of the regenerating family of genes that are associated with beta cell proliferation, protection from apoptosis, and protection from cytokine-induced damage. This suggests that these processes are potentially impaired in ATG7 delta beta cell islets. Next, looking at the pathways that were upregulated at the protein level, we saw an upregulation of proteins involved in response to endoplasmic reticulum stress, with a concomitant increase in chaperone proteins involved in protein folding, as well as proteins involved in ER-associated degradation pathway. It seems likely that the ER stress induced by the ATG7 knockout plays a primary role in beta cell loss in this model. We also found several of the upregulated ER stress response proteins are involved in antigen production and MHC class I complex assembly. The pathways downregulated at the protein level were consistent with the ER stress induced by the ATG7 delta beta cell knockout, including downregulation of protein translation, mRNA splicing, and mRNA processing pathways. The downregulated proteins associated with the translation pathway are displayed on the heat map on the right. And also here showing the downregulated proteins associated with the mRNA splicing and processing pathways. As I mentioned, due to alterations of proteins involved in antigen production, MHC class I complex assembly, we next asked whether MHC class I expression was altered in the ATG7 delta beta cell islets. To do this, I performed immunofluorescence staining for MHC class I on ATG7 delta beta cell pancreatic tissue. And we found that MHC class I was significantly enhanced in the knockout islets. The mean fluorescence intensity for MHC class I islet expression is quantified on the right, showing this increase in expression in ATG7 delta beta cell islets compared to control. I also stained for CD45, which is a marker of all nucleated immune cells, shown in red. We observed a significant increase in the number of CD45 positive immune cells in and around ATG7 delta beta cell islets compared to littermate controls. The number of CD45 positive cells within the islet area is shown here on the right. This finding coincided with our transcriptomic data showing upregulated inflammatory response and cytokine production in the ATG7 delta beta cell islets. In support of this data, I also asked whether pharmacological modulation of autophagy in endoseed beta H1 cells affected HLA class I expression. HLA class I is the human homologue of MHC class I protein. I treated cells with vancomycin A1, which is a lysosome acidification inhibitor and thus inhibits the final stage of autophagic degradation, or cells were treated with C381, which stimulates lysosome acidification and function. The diagram on the left shows where these compounds act in the autophagy pathway. Then on the right is a immunofluorescence staining to show that the treatments are functioning as expected. The lysotractor signal is ablated by the baflomycin treatment, whereas the lysosensor signal is enhanced with C381 treatment and the reduced pH as expected. The cells are treated overnight with either baflomycin, C381, or vehicle control, and then prepped to analyze HLA class 1 expression by flow cytometry. Shown here on the left is a representative sample expression distribution from one of my flow runs. And on the right is quantified the median fluorescence intensity plotted as fold change compared to the vehicle control, which is indicated by the red dashed line. I found that the late-stage autophagy inhibitor, baflomycin, increased HLA class 1 surface expression, whereas stimulating lysosome function actually decreased HLA class 1 expression on endo-C beta cells. As I mentioned earlier, our lab found evidence of lysosome dysfunction in beta cells in people with type 1 and in individuals at risk for developing the condition. This could be a mechanism contributing to enhanced HLA class 1 expression on beta cells that is seen in type 1 diabetes. The primary factor of HLA class 1 the primary driver of HLA class 1 hyperexpression in type 1 diabetes are cytokines, including interferon alpha. And the type 1 interferon response is thought to be associated with events that trigger autoimmunity. So I was interested whether autophagy modulation following interferon alpha-induced hyperexpression would affect beta cell HLA class 1 expression. To test this, I treated NOC beta H1 cells for 24 hours with interferon alpha, then the treatment was washed out, and the cells were treated with followed with a treatment of either baflomycin or C381. Once again, I am showing a representative sample distribution plot from HLA class 1 expression from one of my flow runs on the left, and on the right is quantified the median fluorescence intensity plotted as fold change compared to interferon alpha treated alone, represented by the green dashed line. What we found was that baflomycin maintains the hyperexpression of HLA class 1 induced by interferon alpha above vehicle treatment following washout, whereas stimulating lysosome function with C31 accelerates the disappearance of HLA class 1 following interferon alpha treatment. So it appears that impaired autophagy could exacerbate the effects of interferon alpha on beta cells, which we are following up on with future experiments. Lastly, we wanted to see whether loss of beta cell autophagy altered immunogenicity compared to litter-mate control islets. To do this, HEG7 delta beta cell or control islets were isolated, dispersed to single cells, and islet cells were co-cultured with splenocytes from BDC 2.5 T cell receptor transgenic mice that contain diabetic CD4 positive T cells. The cells were then co-cultured for 24, 48, or 72 hours, and supernatant was collected, and interferon gamma production was measured to assess T cell activation. We found that in the presence of islets with defective beta cell autophagy, there's sustained elevation of interferon gamma production above or in line with interferon gamma production induced by the positive control treated with CON-A, an activator of T cells, and above that of litter-mate controls. Furthermore, litter-mate control islets treated with baflomycin were also shown to have elevated interferon gamma production. We also obtained strikingly similar results when we performed the experiment with a dilution of islet cells. This demonstrates that dysfunctional beta cell autophagy appears to enhance T cell activation and is likely making the beta cells more immunogenic. In summary, we have shown that dysfunctional beta cell autophagy induces ER stress and enhances MHC class 1 presentation. This coincides with enhanced immune cell recruitment to these islets and increased IL-6 and TNF-alpha cytokine production from islet cells and/or immune cells recruited to the islet. We also show enhanced BDC 2.5 T cell activation when beta cell autophagy is impaired. It's possible that alterations in protein degradation and antigen presentation induced by impaired beta cell autophagy could also be leading to neoantigen formation, which we are interested in following up on in future experiments. Overall, we conclude that defects in beta cell autophagy appear to make beta cells more immunogenic and susceptible to immune attack and destruction. In the future, we would like to determine the effect of stimulating or inhibiting the autophagy pathway on the response to interferon alpha and beta cells, including whether it affects the level of activation induced by interferon alpha. Next, we want to perform mass spectrometry on lysosomes from beta cells with impaired autophagy to assess for buildup of misfolded or modified proteins and peptides that could serve as sources of neoantigens to enhance beta cell immunogenicity. We also want to assess the secretion profile of human beta cells under condition of impaired autophagy in combination with cytokine treatment to get a better understanding of how this pathway may affect beta cell immune cell dialogue. Lastly, we are evaluating the effect of impaired beta cell autophagy on immune cell chemotaxis through co-culture migration assay. With that, I would like to thank my lab and my mentor, Dr. Lemon, for her guidance and mentorship on this project, past lab members, Sharanya and Justin, for all their work on the project, and current lab members for all their helpful input and feedback. I want to thank our collaborators at IU's Diabetes Center, Dr. Piganelli and Dr. Roy, for their help in performing the splenocyte co-culture experiments. I also want to thank the IUSM Translation Core and Islet and Physiology Core for their contributions to this work. And I would like to thank my committee members for their input and our funding sources. And lastly, I want to thank everyone at the Indiana University Center for Diabetes and Metabolic Diseases. There are too many people to list individually, but I really appreciate your willingness to give of your time and resources to help better my work and improve my ability as a scientist. My favorite part of doing my PhD training in the Diabetes Center has been that collaborative atmosphere and everyone's willingness to help each other both inside the lab and in life in general. And thank you all for watching.